that's another issue that for patients who are on long-term ibrutinib therapy, one of the ASH abstracts was talking about the pharmacovigilance. So when you're looking at in real life for patients who are on this long-term treatment, there are a lot of patients who may start new medications or have already been on certain medication that can cause potential drug interaction with ibrutinib. So that is something that the clinician have to keep a keen eye on to make sure that you're screening all the patients for all those medications.
It was an impressive amount. You're right. They looked at almost 100 patients who were started outside of the trial. And 2/3 of them at some point were on a medication,
that
most of the time that could have caused their levels to go up and in a few cases to go down. And of course, those patients are excluded from the clinical trials, right? But in the real world, as you just said, they're not excluded. And so that, I think that was a very striking abstract to note that 2/3 of patients, were getting another medication at some point that could affect their levels.
So are there medications that we need to know about, that our audience perhaps needs to know about, that they should avoid in combination with ibrutinib?
I'm sure there are, but I don't remember what they
There's
are.
quite a few.
Where would you go to look at that list of medications?
Yeah, the main concern is with the antifungal and some of the antibiotics and some of the antidepressant. However, even if I agree that the information from that abstract is very important, we do not have a way to measure ibrutinib level. And that's something that we can do with imatinib. So we don't know really if this interaction that is both with the decreased level or increased level is something that carries out in the
I think the rough guidelines have suggested that it's that if it's
a moderate CYP3A4 inhibitor, then you decrease the dose from 420 milligrams to 140.
Right,
but
and if it's mild or whatever, you just decrease it one dose
and you try to
level.
avoid using strong CYP3A inhibitor or inducers.
Probably somewhat empiric, but it's probably better than nothing.
But it would be nice to have the ability to measure drug levels.
So fortunately, though, the therapeutic index is wide enough that the abstract also reported really no negative sequelae of the drug-drug interactions.
What's unfortunate, though, some medications may actually lower the effective dose, and that's of concern because for some reason, patients are told to take 3 tablets a day, and why 3 tablets and not just 1 tablet? In some cases, patients are advised to take only 1 tablet as a form of kinder, gentler therapy, thinking that this might be a way of easing into the therapy, and some patients may be wanting to be maintained on 1 or 2 tablets, and I think this is ill-advised. And if you start taking drugs that can interfere with the drug's level, including going down, there is a danger that you may not have the receptor occupancy for the enzyme BTK, and that's going to be only a partial block on BTK. And one wonders if this is going to be a recipe for resistance over time.
Right, it really is a threshold effect, and you really have to be above that 85% inhibition to have any benefit.
Right, and I think Rick's point was very good because in the original phase 2 trial of ibrutinib, 480 was the dose used because that was predicted from the phase 1 to provide complete occupancy of BTK. And I think reasonably at the time, the thinking was, what if there are off-target effects that contribute to response? And I think there may be with many drugs. So there was a second dose that was in a cohort, which was 840. And the bottom line is that the efficacy was the same, which more or less validated the assay. It wasn't better than 420, and it was not really much more toxic. So I think Rick's point is good. And it speaks to Tom's point. I'd probably be a little more worried about reducing levels than having them go up. Now, the good news is in that abstract, there were only 4% of the patients that were on drugs that reduced levels. So that the majority of them were on drugs that would make the level potentially go a bit higher.
Well, there is some concern because no drug is really completely specific. And in the case of ibrutinib, you do target other enzymes that require higher levels of drug to target those enzymes, and some of them are involved in T-cell biology and the ability to mediate ADCC, perhaps. If you have too high a level, you're going to get more off-target effects, including effects on the platelets and what have you. So I think having more of the drug could lead to higher rates of complications.
I think that's a good point. It might not be the inhibition of BTK itself that would do it, but some of the other effects, yes.
So I think one of the important things to actually remark on are those agents which have been identified to actually diminish the levels of ibrutinib. So we certainly talk about the anti-seizure medicines, but most importantly, St. John's wort is associated with diminishing the level of ibrutinib, and I think it via the induction of the CYP3A4 system. So making sure patients aren't taking, potential remedies that would be including St. John's wort and alternative medicines is just very important for everyone to discuss with their patients on ibrutinib.
I think that's a great point because a lot of times now when you get a list that the nurse might take of concomitant medications, they're really only asking them about prescription medication. So I think that's a really good point, Rick.